My name is Noel Caplice. I'm Professor of Cardiovascular Sciences in University College Cork in Ireland. My talk will be on the role of precursor cells or progenitors in atherosclerosis and vascular injury. Over the last decade, we've been increasingly interested in the role of stem cells in cardiology, and particularly precursor cells that come from the marrow. And our group has really focused in the last decade on the role of a specific subset of marrow cells called smooth muscle progenitor cells. And what we found in that time is that initially, unknown to the scientific field previously, was that there are circulating cells in the blood that become smooth muscle cells. Now, smooth muscle cells are the major functioning and structural component of the arterial system. And so we thought there was two really exploitable perspectives on this. One was that we had a ready-made source potentially of expanding smooth muscle cells and creating arteries outside of the body. And secondly, that we had a novel and potentially targetable source of cells that could be modified to alter atherosclerosis. And so we've set about a number of studies, one to prove in human beings that these cells exist, and they provide approximately 10% of the plaque volume in an adult who has atherosclerosis. Why is this important? Because they're situated in key areas within the plaque. One, they're involved in angiogenesis or microvessel formation within the plaque, and also they're involved in the adventitia, which is really the source of blood supply, metabolic control, and also nutrient control of the vessel wall as the plaque grows. And so we've been able to, through a series of transgenic studies, show in mice that if you ablate these cells, essentially you can block plaque formation. And also we've identified one chemokine receptor on a macrophage, which is the key molecular switch which alters a circulating macrophage cell into a smooth muscle progenitor cell. Why is this important? Well, in one area, if we use a simple antagonist that we've developed with a drug company, we can actually block re-narrowing within stents, which is a big problem with stenting. It's called in-stent restenosis. And in the second area, we can target this angiogenesis by actually blocking this process. Now, this is a double-edged sword, because what we find is that if you do try to block angiogenesis in this way, the microvessels become more leaky. But I think this gives us a crucial insight potentially into what causes plaque instability. So in summary, we really have three key areas that we're interested in. One, which is circulating smooth muscle progenitors, the role of those cells in providing an external source of smooth muscle cells which can be used for tissue engineering. Secondly, a key structure-function interaction between a well-known cell, the macrophage, and smooth muscle cells, which probably determines, at least in part, the stability of a plaque, whether it will rupture and lead to a heart attack or whether it will become stable. And finally, a key molecular chemokine, which is the CX-301, which may be key in targeting plaque growth and particularly in-stent restenosis. Increasingly, at least cardiovascular research is becoming fragmented. So we have meetings that are specifically related to basic research, meetings that are translationally oriented, or meetings that are purely clinical. And I think what we have to do is realize that a lot of clinicians want to know what's going on potentially in the next 2 or 3 years. What are they going to see coming down the track? And I think meetings like this can fill that gap that's not necessarily well catered for by the big cardiovascular scientific meetings because really of pressure, and that is because of the fragmentation. There's a lot of pressure in terms of clinical trialists to simply have a meeting for that. There's a lot of pressure from basic scientists saying we just want to speak to each other. But really, for at least in my own experience, because I'm a clinician, I'm also a basic researcher, you need to have dialogue between both. And people need to get inspired by what's going on in the basic research field and take some of those ideas into their own clinical practice.